today's talk we have Yuhua, who hopefully all of you know from many emails. But today she's going to talk about her research, and her research is on why brains say stop. So I hope our brains don't stop with all the mind-blowing science that we're about to see. Over to you.
Okay, so in case anyone of you who don't know me, my name is Yuhua. I'm a second-year PhD student. So today I'm going to talk about how our brains say stop to memories and actions. So it may sound a bit counterintuitive at first, because normally we would think about how we do things and how we act on things and how our brain processes to allow us to be able to initiate and execute all of that. So why do we need to stop? So imagine this scenario when you're sort of taking a break from your work and you're just browsing random YouTube videos, and then you should really get back to work now, but then you have this up next video suggestion from YouTube. It really makes it very convenient for us to procrastinate a bit more, right? So here there are two things that we need to stop. First off is one of the images was gone. Okay, so first off, you need to basically resist the motor action to click your mouse into this next video that YouTube has suggested you to watch. And then the other component is that you need to sort of stop to suppress the thought of, okay, I'm just gonna watch this one more video and then I'm gonna get back to work. So that's like an everyday scenario where we need to stop a motor action and a thought in our mind. But it probably doesn't do much harm even if you did watch this one more video. But in some other scenarios, for example, when you just experience a traumatic event, or if you're about to do something that will be harmful to yourself or someone else, then it would be really important that you do stop your thoughts or action, because if you did think about it or do something, then it might yield some serious consequences. So, having those in mind, so those are the real life examples. Then how do we study stopping in the laboratory situation? How do we standardize a procedure to have everyone respond in the same way and quantify people's ability to stop a motor action or memories? So in terms of motor action, there's this quite established paradigm called the stop signal task. So basically it's a button pressing task. There are two types of trials in this task. The first type of trials is a go trial. So basically you see a very simple stimulus on the screen, or maybe you hear a tone or something, and your task will be to react to the stimulus and press a button. And as simple as that. But on other trials, the stop trials, soon followed by the initial stimuli, there will be a stop signal that you receive. For example, an audible tone or something like that. And as soon as you hear that tone, you're supposed to refrain from pressing the button. So you can see that there are two differentiated types of trials where you need to initiate an action, and then in the other type you need to basically inhibit this urge of pressing that button, because typically in this sort of tasks there will be more go trials than the stop trials. So basically you form this habitual response to pressing a button, but in a minority of cases you have to inhibit the urge of pressing this button. And then coming to stopping memory retrievals. So this is typically studied using this think-no-think task that my supervisor has developed some years ago. So basically in the think-no-think task, like in the stop signal task, you have trials where you need to think about things and initiate memory retrieval, and in some other trials you need to refrain from retrieving the memories that you've learned. So typically in this task, you have three types of stimuli. So recall, suppress, and baseline stimuli. So basically in the recall stimuli, you need, well, I I should go through the task. So basically, there are 3 phases in this task. In the first phase, it is a study phase. In the study phase, you typically learn Q target pairs, and participants are trained with these pairs to a certain criteria before they can proceed to the later phases, because you need to know these things to be able to recall them or suppress the recall. And then in the second phase, the think-no-think phase, the recall items will show up in green, meaning that the participants are supposed to recall the target item that were associated with the Q. Whereas the suppress items, the Q will be shown in red, and then the participants are instructed to try not to think about the target item that were associated with the Q. So here you can see that for some items, they're repeatedly retrieving the target, whereas for some Qs, they're repeatedly suppressing the target. The baseline items are simply not shown in this phase because they're baselines. And then finally, there will be a surprise memory test. So participants didn't expect this, but in the end, there is a test to measure how people's memories vary depending on which type of stimulus it was. And typically, what we observe is that relative to baseline, there will be a facilitation of recall in the recall items because they've repeatedly trained themselves to retrieve those memories. Whereas for the suppress or the no-think items, there will be a significant decrease in the level of recall because during the think-no-think phase, they've repeatedly trained themselves to specifically suppress the targets of those pairs. Right. So this means that people are actually able to suppress memories if they want to, which is quite interesting. So these are the typical behavioral paradigms that people use in laboratory situations to study how people stop memories and motor actions. So what are the typical brain activations then? Because we're talking about the brain today, right? So this is a project that is done by some colleagues in my lab. And so basically what they did was they did both the stop signal task and the think task in the very same participants. So it's a within subject design. So basically they put people into the fMRI scanner and they asked them to do the stop signal task and then the think task, or the other way around, to see what are the brain activity that are stimulated by these 2 different tasks. And what they find is, so here I want to ask a question, like, what do you think they'll find? Do you think they'll find like very different brain activations because it's 2 different tasks, or do you think they'll find very similar brain activations because they're stopping something? Anyone?
Could be similar. Could be similar.
Similar?
Yeah.
Could be different. I'm going to go with different because otherwise it'll be a very short talk.
Yeah, they're the same. But actually, they are quite similar. So in brain imaging, typically we look at data in contrast. So basically, for example, in the stop, greater than go contrast, we're subtracting brain activations associated with the go condition from the brain activations associated with the stop condition, so that you know that these activations are stop specific. And same in the no-think greater than think contrast, these activations would be no-think specific. So basically you can see that stopping a motor action and stopping a memory retrieval seem very, very similar to each other, which is very interesting. And then when we and also they did a conjunction analysis, basically looking at what are the overlapping brain activations that occur in both of these tasks, and this is what they found. So I made this brain kind of transparent because these activations are kind of deep in the cortex, and it would be impossible to see if I did something else. But wait, where did it go? So I'm not sure how familiar are you with the brain. So basically, so let me just go through some very simple ways that people describe brain activation. So basically, here is my brain and you have yours. If anything in the front, we call it anterior. Anything in the back, we call it posterior. Anything in the top is superior, and anything in the bottom is inferior, which is quite straightforward. And then, so here we're looking at the cortical activation. So basically, so you know that the brain has the cortex, and then below the cortex, there's some other complicated structures. So the cortex is generally divided into 4 different lobes. So this over here is the frontal lobe. So the frontal lobe is generally involved in high-level cognitive control. So for example, attention, executive planning, making decisions, and motor control and stuff like that. And so here, this part, slightly to the back, is the parietal lobe. So the parietal lobe interacts with the frontal lobe a lot for attention orienting and stuff like that. And it's also involved in spatial navigation and some memory. And this, in the very back, is our visual cortex. So basically, basically you're seeing me right now, and this information is sent all the way back to your to the back of your brain for visual processing. And this over here is the temporal lobe. So the temporal lobe has a lot to do with language comprehension, auditory processing, and the medial temporal lobe, for example, especially the hippocampus, is very important in memory formation. So some of you might know that if you don't have the hippocampus, if you have it removed because of epilepsy or something like that, you will simply lose the ability to form new memories. So it's very important. And this little bit over here is at the bottom is the cerebellum. So here you can see that basically when we do the conjunction, there are a few clusters that specifically stand out. So there are quite a few of them in the frontal lobe. So as I said, the frontal lobe is very important in executive functioning and planning. So this little cluster up here is the area that is involved in motor control. Specifically, it's involved in volition, so voluntary control of action. But maybe if it's showing up in memory control as well, maybe it's involved in a voluntary control of memory as well. That would be a hypothesis of why it is showing up here. This cluster over here is the what we call the dorsolateral prefrontal cortex. So dorsal is something to the side, and medial would be to the middle. So dorsolateral prefrontal cortex is repeatedly found in memory in stopping memories and stopping actions. So this may be a very important cluster that is involved in controlling or stopping both memory and actions. This little cluster over here is repeatedly found in motor control. So like for people studying the stop signal tasks, they will emphasize that this cluster is really important, or if not the most important cluster for motor control. But you know, since the other ones up there as well, we probably don't want to be that conclusive for now. And then again, this parietal cluster, it may interact with the frontal clusters for, you know, directing attention, or like maybe it's involved in some memory processing, but we're not entirely sure yet. So you have an idea of how it generally look like when we put stopping memory and actions together. But also you may say, this is only one study, right? It could be a single case that we're finding a similarity between stopping memories and actions, or it may be a general effect. How do we know about that? So then it comes to a part where I did a lot of the work with a postdoc in my lab. So what we did is a meta-analysis. Do people know what a meta-analysis is? Yes, no? Okay, so basically a meta-analysis is when you put together a range of similar studies and you want to find out whether something holds true in all of them. So for example, we have a number of studies and we want to see if the overall effect size, like if there's an overall effect size in all of the studies. And we did this for the stop signal task. We did this for the think no think task, and we did the conjunction as well. So now, by the meta-analysis, we'll be able to answer the question of whether the brain maps that we observed for stopping motor and motor actions and memory holds true in their separate domains, and when we do the conjunction analysis, whether the conjunction map we saw just now would still hold when we when we aggregate lots of different studies together. So in terms of fMRI analysis, like there's this very useful tool called the Ginger Ale for fMRI meta-analysis. So again, if we feed a range of studies into it, it will. So basically, the process is we have coordinates of brain activations because the brain is a 3D volume, and you note the activations using the coordinates. So you feed it with the coordinates, and it's weighted by the sample size of this study, and then it goes through calculations and correction for multiple comparisons, and then it generates a map for common activation amongst these studies, like this one up there. So what we find for the meta-analysis, so here my apologies, I need to show you a different type of graph because again the activations are slightly hard to see if it was presented in the other brains. So basically this is the same as all the brains that you've seen, except this is an inflated cortex. So imagine the folds on the cortex. Imagine your brain is a balloon and you inflate it, so the folds will now become flat. And these gray and dark gray and light gray areas would be where the folds lied previously. And the color areas over here are cortical areas that are separated by their anatomical like cellular structures and functions. It's just simpler so that you can easier see how the brain activations localize. You don't have to worry about how different each region is. And again, I had the 4 lines so you can have an idea of how this is as compared to the previous brain. So again, for both, so we did this for both the stop greater than go and the no think greater than think contrasts. The results are pretty similar. So again, you see these frontal clusters and these parietal clusters. So we can say that the similarity in stopping actions and stopping memories are true even when you consider lots of different studies together. And again, we did the conjunction analysis, and this is what we found. So again, the dorsolateral prefrontal cluster, the ventrolateral prefrontal cortex cluster, this parietal cluster, and the volition-related cluster. And if we compare it with the fMRI study, the fMRI conjunction, you can see that these clusters are virtually identical. So it means that, hey, there's really something similar between stopping a motor action and stopping memories. Isn't that cool? Yeah. But having said that, obviously, like there will be task-specific differences because they are different tasks. But by finding similarities between them, it means that maybe there is something central for stopping just anything in general. There may be an inhibition hub in the brain that we're not realizing. That being able to stop is so important in our life that some special networks in the brain are dedicated to it. So Okay, so far we've been talking about the cortex, but at the same time we also had some subcortical findings. So it is a brain, and the color-coded areas are subcortical structures. And again, for both the stop greater than go and the no think greater than think, we find very similar subcortical activations, and these are the only activations that showed up from the meta-analysis. You can see that they're very similar, except in the stop greater than go, there's it's bilateral, but it's fairly symmetrical. So you may ask, what is this activation then? So this activation lies in this structure called the basal ganglia. So the basal ganglia is a set of subcortical nuclei that is traditionally very well studied in motor control. So for example, in Parkinson's disease, and this picture is a very broadly used picture to symbolize Parkinson's disease. So basically, some of you might know that Parkinson's is a neurodegenerative disease, and the symptoms are caused by deficiencies in the basal ganglia system. So basically, within the basal ganglia system, there are different pathways that are in charge for initiating an action, stopping an action, and just overall inhibiting any sort of unnecessary actions. So when you have deficiencies in this system, you could have unnecessary movements or talks, for example, the Tourette's syndrome. I'm not sure if you're familiar with that. And then so that would be when you have more, but when you have less, you would have something like Parkinson's, where you have rigidity in your movement. So like I said, the basal ganglia is traditionally mostly studied in the context of motor control, but more research has indicated that actually it's involved in lots of different functions. For example, depending on where specifically you're looking at in the basal ganglia and which specific areas it is talking to the cortex. So, for example, motor control is only one functional division, and there are also others. For example, visual, emotional, and executive. So we would be like talking about stopping, which is a relatively higher level function. We would be most interested in how the basal ganglia is interacting with the executive networks. So. Again, this is the conjunction for the subcortical findings from the meta-analysis, and we will need to know where exactly this activation is, so that we can relate back to. Well, why is it jumping so fast? Right, so we can relate back to this graph and say, here it is, the activation, and this area of the basal ganglia specifically talk with these cortical regions, and hence we can form hypotheses and say, this is how it is doing the task that we're interested in. And sadly, in humans, such evidence doesn't exist yet. So we looked into some animal research, and I found this paper. It's a tracing paper using monkeys. So basically, they injected dyes into different cortical regions in the monkey brain, and see which fiber tracks these dyes would travel and end up in the basal ganglia. And so I'm not sure how well you can see it. So basically, these are color-coded to show that different cortical regions have concentrated projections into the basal ganglia. And so if we can localize the basal ganglia activation, we could possibly deduce in the reverse direction and say these are probably the cortical regions that are talking with it to achieve the tasks. So you can see that our human brain activation it's quite similar to this yellow-orange region, and this yellow-orange region correspond to projections from the dorsolateral prefrontal cortex, which is this cluster that we find in the meta-analysis. So it could be possible that stopping motor action and stopping memories have this common mechanism lying in the interaction between this specific prefrontal region and the specific basal ganglia region. But of course, the next step would be to find out how it is achieved and whether stopping. So we know that motor control is achieved through the multiple basal ganglia pathways that I briefly mentioned just now. Will controlling memories go through the same pathways, or is it slightly different? And also whether how this system interacts with the task-specific networks. For example, in terms of motor control, how it interacts with the motor cortices, and in terms of memory control, how it interacts with the memory areas, for example, the hippocampus that I mentioned earlier in the talk. So obviously, so in my lab, we're more focusing on the memory side of the story, and it's not really done yet. So that would be the next step for my PhD as well, and I'm really excited about it. And that's it. So in the end, I would like to thank my supervisor, Professor Michael Anderson, and Dr. Taylor Schmidt. He is the postdoc that I've been primarily working with for this project. And I'll be happy to take questions if you have. Yeah.
In the stop-start experiment,
yeah.
How do you tell if someone is or is not thinking about the don't think? Or, you know how they had to not recall one of the words?
Yeah.
How do you tell if they're not recalling that word?
So that's a very tricky thing to do, because basically they're just doing everything in their mind, and we can't read people's mind just yet. So so typically what we do is we run although it's a memory experiment, we run it as an attention experiment. So we would say that when you so this is an attention experiment context, and when you see the green word, you need to draw your attention to the target word, whereas for the red word, you need to focus attention on the red word and not think of anything else. So like anything else would be distractions.
So it's eye movement.
So it's primarily just an instruction thing, and we go through like questionnaires and stuff to make sure that people are compliant. But that is definitely a valid question.
What do you mean I don't understand. How you tell? Is it their eye movement that they're associating with?
There's basically no direct measure of it. If that's what you
What
mean.
about the recall words then? How would they speak the other word? They say the other word?
Originally, like in the very early version, they would. But
They would
then,
say it, okay.
yeah, but then they decided to take that out because they were confused with motor action. Because when you speak, it involves controlling your mouth and voicing the words that you're saying. So now, basically, through each step of the think no think phase, we have a very detailed questionnaire asking people if they've been following the instructions that we're giving.
What are the instructions to recall a word? Yeah.
So basically, am I going to recite the instructions to you now?
Well, how would I recall roach if I was participating in the study?
So basically, the instructions would be now you have the hint and the response word, right? So like basically the hint word will be so some of the hint words will be in green, and when you see green, you're supposed to think of the target word as soon as possible and keep it in mind the entire time when the word is on the screen. Whereas when it's red, you should focus your attention on the red word only and not let anything else distract you.
And then after I tell you if I was able to do that?
Yes.
Okay.
Yes. So like typically the think no think phase will be segmented into different into a few chunks. And for example, after in the middle or after each chunk, we'll have this questionnaire to ask people if
Do you know how long the chunks are?
a few minutes.
How many words would you get through, or how many associations would you get through?
So typically there are 60 something associations that people learn, and they're pretty good at it. So basically in the study phase, we would train people with these pair associations to at least 60% of accuracy.
And do they know beforehand if they're going to be a recall word or a suppress word?
So all words. So basically, what you're seeing here is how they would see it. Like in the learning phase, they would see everything the same. They wouldn't be able to tell which type of words they are. And then in the think no think phase, they see green or red or not see it at all. And then because it's an attention task, so they're not supposed to expect a final test in the end. And so hopefully that wouldn't interfere with the think no think phase, because if people expect to be tested in the end, they probably just won't perform the suppress instructions anymore. And then they have a final test to see how their levels of recall vary because of those different conditions.
I was just wondering, what do you think might be the difference between memory and attention?
Memory and attention.
Yeah, so you just said it's more of an attention task. Like, what other parts of the brain is memory involved?
So, memory is primarily in, well, I guess it depends on the specific types of memories that you're talking about. So, like, if you're talking about memory formation, then it would be hippocampus and the mediotemporal lobe related area. But then if you're talking about long-term memory, now people like, or schematized memory, people think that they're more transferred into the neocortex. So like, if you say, well, I think it's almost impossible to say that there is one region in the brain that is in charge of any one of the functions, because like it could people used to have that view that the brain is pretty segmented, and then each region would have its designated function. But now, since we have more understanding of the brain, we know that the brain works in networks. So for each function that you're performing, there will be a network of areas that are collaborating to achieve it.
So what networks, then, the ones you've described here, are the networks of the particular networks of memory that your work is looking at? Whereas there could be other networks of memory that your work isn't looking at.
So in terms of stocking memories, we're specifically looking at the DLPFC hippocampal interaction. So in some previous research, people have done analysis to show that there is so basically the role of these regions are that the DLPFC with the dorsolateral prefrontal cortex would send top-down signals to the hippocampus, where local inhibition would happen, so that the hippocampus will sort of inhibit itself from calling in the memories. If that makes sense.
Yeah.
And then so we're running it as an attention network, but essentially what people are performing is still a memory task. So there's also this paper looking at how this specific paradigm can be tapped into the attention network, and because we typically see the frontal parietal attention network showing up in this task. So as I said, that might be one of the functions of the parietal activation, and that would be a component to the that would be definitely a component to the task. But in terms of the memory, we're more specifically looking at the prefrontal hippocampus interaction.
Has anyone done any studies on memory? Oh, sorry, on emotion, and how emotion might affect performance in such memory tasks?
Yeah, so some of these, so basically...
So actually, I wanted to go back to your slide with the picture of the brain that had like emotional areas and all of these, and did you find that the emotional areas were in any way activated?
You mean?
The other, no, it's later, I think.
So, no?
No, we might never find it now, don't worry.
Because those are the only brains that have.
No, yeah, it was the one, and it had a nice pretty one with full areas
Ah.
of emotion and that one.
That one. Okay. Yeah. So, so what was your question again?
How did emotion? Has anyone done any studies to look at how emotion would affect memory in these sorts of attention tests?
Yes. So, like the example I gave was a word pair task, but like people have also used emotional stimuli, for example, emotional pictures, to look at to see whether people's ability to inhibit memory would vary between emotional and non-emotional stimuli. And there are also people who did experiments on autobiographical memory to look at if people to see if people are able to suppress their own memories, which might be a more realistic task. Yeah.
Did they find anything that, like, how would you control for memory for emotion in your tests?
So typically in psychology experiments we use standardized databases. So basically there are databases of pictures, for example, that have standardized ratings, and we would basically select pictures in a range of level of valence, that is emotionality and arousal. So we will hope to match the level of arousal and valence in between different groups of pictures, so we know to control for variability.
Okay.
Yeah.
Has anyone tried using the imaging data to classify which words were being shown? So like using and then you could maybe get a picture of which word pairs tend to produce more predictable results in people.
That's
Like I
really
find steam train easier to remember than roach or deal, because I don't know what the connection is between roach and deal.
Yeah. I don't think anyone has done it with this paradigm, but there was a recent paper using MBPA, like multi-pattern. What is it again?
Multi-voxel
pattern analysis. Yes. So it's basically it's a multivariate analysis looking at pattern similarities and see if, like what Matthew said, like if you could train a localizer or something and see what are the pattern of activity when a specific word shows up, and then when it later shows up, will the level of activity increase or decrease depending on the condition that it was in. So that was a very good idea. There was a recent paper using a slightly different paradigm with MBA, and I think they did find something. Yeah, I can't remember for sure.
It's like, unless two things could go into it, but like if you try predicting which word was being shown for all different people, and then for one of the people the predictions are absolutely shot, then either that person is thinking of the word and doing something in a very different way than everybody else, or they're not thinking of the word. So it might be interesting, like matching that with the questionnaire results to see like if they've admitted
that would definitely be a more objective measure of whether people are thinking or not thinking of the words. So now mostly I think what people do is to have an intrusion rating after each item. So like basically in the think phase after each red or green item, they ask the participants to subjectively rate how much the target item was brought to mind. But again, that's still a subjective measure, and if we could use multivariate stuff to actually quantify how much these items are brought back to mind, then it would be really good thing to do.
So you are talking about which part of the brain are controlling us to stop thinking of something or doing some action. And what about start to do something? Do you think it will evoke the same part of the brain?
So again, that'll depend on what you're doing. But in terms of the basal ganglia, for example, as I said, the basal ganglia have three. Well, now people know that the basal ganglia system has three different pathways. So there's a hyperdirect pathway that performs sort of a global inhibition. So if I'm standing here, I'm not doing anything unnecessary. If there's something wrong with the hyperdirect pathway, I could be just doing random stuff right now. And then there is the direct pathway that allows you to initiate chosen actions. So for example, if I want to grab something over here, I know that this is the best way to grab it because of the experience and because the direct pathway has allowed me to do that. And then once something is achieved, the indirect pathway will shut this down and I will no longer be reaching forward. And then the system will shift to something else, to the next step of the movement that I should do. So that's in the motor control area. And so now we don't we don't really know how the basal ganglia system is interacting with the memory networks, and that's what we're trying to find out right now. So the hypothesis would be that memory control would go through the same pathways as motor control. So like basically there would be the hyperdirect pathway that's sort of performing global inhibition, and then direct pathway interacting with the memory system to initiate memory retrieval, and the indirect pathway to interact with the memory system to stop memory retrieval. But we'll need to find out about it.
Has anyone compared the sort of the mental, well, like the stopping, just the whole general stopping thing in general with people when they're meditating? So I remember reading something that they got some monks and got the monks to meditate and did loads of EEG analyses. Has anyone compared those two data sets?
I'm not sure. Yeah, but so meditation and mindfulness is apparently a very hot area in recent years, and a lot of people are doing mindfulness research, and there are hypotheses saying that like if you do a lot of meditation and mindfulness training, you may be better at controlling thoughts and orienting attention and stuff like that. So I did a behavioral pilot earlier this summer using a similar paradigm, and I had a mindfulness questionnaire in the end. I didn't find anything, but some of my colleagues they used the same they used similar paradigms and questionnaires, and they found maybe there's something, and that was why they suggested me to have the questionnaire. But again, it's a subjective measure because it's a questionnaire, and it's so I'm not sure about that.
But I mean, in terms of the actual areas of the brain that were activated between the two studies, like in the way that you compared all the separate studies and looked at the similarities.
I'm not aware of studies like that. Maybe they exist.
Wasn't the whole point of meditating not doing anything? Stopping doing anything? I guess you're suppressing doing.
Yeah, suppressing anything from happening.
And so people good at meditating should be really good at these tasks. Yeah. Like I have my next experiment, so look at it. People who think you're very good at meditating.
Just email the demo. She's got it now.
Cool. So if there's no other further questions, and if there are, you can always ask her. You're staying around, right?
Yeah.
For a little bit now, yeah. So then we have wine and cheese set up for you in the other room. So let's thank you again for the great talk.